Now we'll today discuss on a topic from the paper C4, paper C4, cell biology paper, and the unit is structure and function of lysosome. Okay, so I think in our previous class we have learned about the structure and function of Golgi bodies. Okay, in my part you are assigned with the structure and function of Golgi bodies and lysosome. Okay, so now today we will discuss about the lysosome. So before entering into the topic, what we will learn from this class today? Okay, so first the definition of lysosome. What is lysosome? Okay, so that is the definition of lysosome. History, how the lysosomes are derived. Okay, who coined the name lysosome or like that. Okay, then occurrence, where the lysosomes are found. Okay, then structure of lysosome, kenekwa hoy dekhibole, how it looks morphologically as well as internally. Okay, and then enzymes of lysosomes and function of lysosome. Okay, enzymes of lysosome and function of lysosome. Okay, acha, so in our previous class when I am asking about this lysosome, I remember someone has said that term which we use for the lysosome. Ki bole ko ami lysosome? What is lysosome called as in general term?
Suicidal bags.
It is called as suicidal bag. Okay, it is called as suicidal bag. Kele koisile, why it is called suicidal bag, we will discuss in our today's class. Okay, why the lysosome is called as suicidal bags. Okay, acha. Now coming to the definition. So lysosomes are spherical organelle that contain enzymes. Okay, these terms are important. Okay, they contain the enzymes. Okay, they contain the enzymes, and which kind of enzymes? The enzymes are acid hydrolases. Okay, the enzymes are acid hydrolases. Now acid mean we know that pH is below 7. Okay, acid hydrolases. What does this word mean? Hydrolases. Hydrolases mean the enzymes. Okay, which can act on or which can digest some particle. Okay, which can break down some particle in presence of water. Okay, which can break down some particle in presence of water. Those things are called as hydrolases. Those enzymes are called as hydrolases. Okay, and here what kind of hydrolases? They contain the acid hydrolases. Okay, they contain the acid hydrolases. After that, they break up food, so it is easier to digest. Okay, we will discuss this point in our function part. Okay, so they break up the food, so it is easier to digest by the cell. It helps the cell, actually it helps the cell to facilitate the process of digestion or absorption. Not digestion, it is absorption. Okay, they are generally found in, they are common in animal cell. They are common in animal cell, and very few are found in yeast and plants. It is discovered by D. Diouf in 1955, and he also coined the term lysosome. Okay, then the first electron micrography of lysosome was carried out by Novikov, and in 1974 D. Diouf got the Nobel Prize for the discovery of this lysosome. Okay, so that point is discussed. The next point is discussed here. The lysosomes are common in animal but very rare in plant. Okay, and the size of lysosome is varied. Okay, it varies from 0.1 to 0.8 micrometer. Okay, 0.1 to 0.8 micrometer, and they are larger in phagocytes. Now what is phagocytes? Phagocytes are the WBC. Okay, which can engulf the foreign particle. Okay, phagocytes are the WBC which can engulf. Engulf mane buzi poa na? Engulf mane khai dia. Like this, if this is a cell, okay, if this is a cell structure, okay, if this is a foreign particle, then to take inside this thing is called as engulfment. Okay, so this is the engulfment. So the WBC generally perform this function of engulfment. Okay. Now coming to the occurrence. It is discussed a little bit here, that it is present in the animal cell. Okay, but very few in plant cell. Most abundant in macrophages. Again macrophages. Here macrophages again means the WBC. Okay, they are abundant in WBC, which perform digestive function, because the WBC has to kill the foreign particle. Okay, foreign particle, to kill the foreign particle they must break down the foreign particle into several pieces. Okay, and therefore they are abundant in macrophages. Then found in cell organ which are associated with enzymatic action, like there are some cells in our body which perform this enzymatic action. Okay, and they are more in those type of cells. For example, liver, pancreas, thyroid, intestine, we know that now. So liver secrete not enzyme, but liver secretes some digestive juice. Okay, then pancreas secrete the enzyme. What are the name of the enzyme secreted by pancreas? Anyone? Eta question. What is the name of one enzyme?
Insulin.
Oh, insulin. No, insulin. Insulin is a hormone. Yes, insulin is a hormone. I am not saying about the hormone thing. I am saying about the enzyme. Insulin and glucagon are the hormones secreted by pancreas. So, anyone now?
Trypsinogen.
Navarita has said the trypsinogen, and then someone is saying another one. Pancreatic what?
Pancreatic amylase.
Pancreatic amylase. Okay, acha. So see, trypsinogen is a inactive form. The enzyme is trypsin. Enzyme is trypsin, but pancreatic amylase is the correct one. There are a number of enzymes like pancreatic amylase, pancreatic lipase, trypsin, chymotrypsin. Okay, so these are secreted by pancreas. So the organ which are associated with this enzymatic secretion, they also contain more amount of this lysosome. Okay, and they are generally absent. They are absent in bacteria. Now structure. If we go for the structure, see the lysosomes are very simple in structure. They are very much simple in their structure. They do not have so much of complex cell structure like that. Okay, for example, lysosomes are globular and diameter is 0.2 to 0.8 micron. In our previous slide we get 0.1 to 0.8 micron. That is also okay. Okay, their range is 0.1 to 0.8 micron. Under the electron microscopy, lysosomes appear dense. Okay, they appear dense and finely granular. Okay, they contain some granule inside their cytoplasm, not cytoplasm, inside their structure. Okay, then lysosome is essentially a sphere of lipoprotein membrane containing the enzymes. The membrane of lysosome is made up of lipid and protein. Therefore, it is called as lipoprotein. Then the membrane insulates the enzyme from the rest of the cell. Okay, since the lysosomes are present. Okay, since the lysosomes are present inside the cell. Okay, and the membrane only separate the whole lysosome from the rest of the cell. If the enzymes are released from the lysosome, they can digest the cell. Not in every cases. There are certain cases like if there is any injury, the cell must have to die. Okay, the cell has some injury, the cell has to be died. Okay, then the lysosomes enzyme are released, and then the cell get digested. Okay, and therefore it is called the suicidal bed. Then lysosome membrane, it is bounded by a single lipoprotein membrane. It is not bilayer, it is single lipoprotein membrane. Then the membrane is impermeable to substrate of the enzyme contained in the lysosome. Okay, so that means this point means that the enzyme cannot cross the membrane. Okay, the enzyme cannot cross the membrane, and certain substance called levelizers causes instability of the lysosomal membrane, leading to release of enzyme from the lysosome. When there is a need, the lysosome enzyme can be released. Okay, due to the flexibility of the membrane. So this is the structure of lysosome. This is the structure of lysosome. This is the whole cell. The blue diagram is the first blue diagram where the cursor is a whole cell. Okay, and this green one is the lysosome. Okay, so if we zoom the structure of the lysosome, we can see here it has a membrane. It has a membrane which contain the which contain lipid and protein, lipoprotein membrane. Then, and these are the enzymes which are found inside the lysosome. Okay, so it is a very simple structure. In structure, you just have to mention these 2 points only. Okay, nothing else. Now this point is important. Polymorphism of lysosome. Okay, polymorphism. Now we are aware of the term polymorphism. Kak koy ami jano polymorphism. So what is polymorphism? It is the different structures of the same organism. In our last semester, we have discussed a lot about the polymorphism. Okay, so here lysosome also have different structures. Okay, depending upon their function. Lysosomes are again divided into different structures. For example, primary lysosome or storage granule. Okay, then heterolysosome or digestive vacuole or phagolysosome. Okay, we will discuss one by one. Just initially get the points here. Next, autophagic vacuoles or autolysosomes or cytolysosomes, and finally the residual body. Okay, these are the 4 different types of lysosome which are visible inside a cell. Okay, now so let me do one thing here. The whiteboard is visible, I think, na? Whiteboard is visible. Okay. Now see. Now let us consider this is a cell. Okay, this is a cell. Now inside the cell, first let us consider this is a lysosome. Let us consider this is a lysosome, which is the primary lysosome. Okay, this is the primary lysosome. Now why it is called as primary lysosome? This is the primary lysosome. Now this is the primary lysosome. Why this is called primary lysosome? Because inside the lysosome, they just contain the enzymes. Okay, they just contain the hydrolytic enzymes. Okay, hydrolytic enzyme, and their function is storage only. Okay, just here come ki they store the hydrolytic enzyme. Okay, they store the hydrolytic enzyme. Okay, that is their function only. They store the hydrolytic enzyme. Okay, nothing else, just storage function. Okay, therefore those are called as storage vacuoles too, or storage lysosome too. Okay, just lysosome is there. Inside the lysosome, the acid hydrolases are there. Different acid hydrolases are there. Okay, so this is the primary lysosome. Now what happened if some phagosomes, these are called as phagosomes or pinosomes. If this is contains solid food, then it is called as solid substance, then it is called as phagosome. If it contains liquid, then it is called as pinosomes. Okay, now these phagosomes and pinosomes, they enter into the cell. Okay, they enter into the cell by the process of called endocytosis. Process to hold endocytosis. That means the entry of these vacuoles inside the cell. Okay, that is the endocytosis. Okay, now whenever these vacuoles enter, these vacuoles are then get in connection, or they get attached with this lysosome. Okay, then they are attached with this lysosome, and now this lysosome contain the enzymes. They contain the enzymes along with the pinosome or phagosomes. Okay, they contain the enzymes along with the pinosomes and phagosome, and now this will be called as secondary lysosome. Okay, this will be called as secondary lysosome or heterosome. Okay, secondary lysosome. That means, see, ultimately what is it? It is the first primary lysosome itself. Okay, initial one. It is the initial one, but with some phagosome or pinosomes. Okay. Now what is the function here? The function is that it can now digest that particular thing, either solid or liquid. Okay, either solid or liquid. It contains these enzymes. It contains the different enzymes, lysosome, and this lysosome enzyme now can digest this thing. Okay. Acha. Now digestion hoigol. Okay. Now digestion completed. If digestion completed, then if digestion completed, then what happened? Then the lysosome. Now the lysosome contain only the, along with enzyme, it contains some residual substances. Okay, it contains some residual substances, and therefore these are called as residual. These are called as residual lysosome. Why residual lysosome? Because it contains the undigested matter now. Okay, it contains the undigested matter now. Digestion hoi gol. After digestion ki hol? Jikhini roi gol, the undigested matter or indigested thing are now remain inside the lysosome, and this will be called as residual lysosome. Okay. Then what is then the third one? That is the autophagosome. Okay. So there are some lysosome which can burst out or which can engulf the cell organelle, its own cell organelle. It might be mitochondria, it might be endoplasmic reticulum, okay, or it might be the whole cell. Whenever there is a time of injury or there is a time of, because see, the mitochondria get aged. Okay, with time mitochondria efficiency komi jai. Okay, then this mitochondria has to be removed from the cell. Okay, and this will be done by this thing. Okay, this lysosome, and therefore these are called as autophagosome. Okay, am I clear? Am I clear?
Yes, ma'am.
Okay then. So these things are here written just here. So primary lysosome is the newly formed lysosome with hydrolytic enzyme only. It only contains the hydrolytic enzyme, and see, the pH is always acidic. Okay, the pH is always acidic because it contains the acid hydrolases. Okay, then primary lysosome where are in attachment with the phagosome or pinosome, they are called as secondary lysosomes. Then residual body, these are the exhausted lysosome with residue only. And finally autophagic vacuoles, lysosome, here the spelling is wrong, lysosomal, it should be lysosomal enzymes and organelle from cell's own cytoplasm. Okay, so these are therefore auto means own, phagi means to eat. Okay, to eat. Okay, therefore these are called as autophagic vacuoles or autolysosomes. Okay, so this is the diagram what I have drawn there. So this is the food particles. These are the food particles, either phagosome or pinosomes. Okay, they are entered to the cell by the process of endocytosis. Then this is the primary lysosome. Okay, this is the primary lysosome only. So they contact and fusion of the vesicle membrane, and they form the secondary lysosome. And this is the digested molecule get diffused across the membrane of the cell. The digested material get diffused, and the remaining one is called as residual lysosome. Now these are the different enzymes. These are the different enzymes we can find in the lysosomes. Okay, first one is the called as nucleases. Which nucleases? Say nucleases. That means they can act on the nucleic acid. Okay, they digest the nucleic acid or they can act on the nucleic acid. So here it includes the ribonuclease and deoxyribonuclease. So the name itself says ribonuclease and deoxyribonuclease. So can anyone tell me what is ribonuclease or what is deoxyribonuclease? Just your idea about this thing, ribonuclease and deoxyribonuclease. Anyone? Since nuclease, I have said it act on the nucleic acid. It can break down the nucleic acid. So what is the ribonuclease then? Say Kusum.
Maybe a ribonuclease, which is the nucleus which is present inside the ribosome, and deoxyribonuclease, the nucleus which is present in DNA. Maybe.
No, not like that. Not like that. Because see, Kusum, we are not talking about the enzymes of ribosome and DNA. We are talking about the enzyme of lysosome. Okay, these are enzymes present in the lysosome. Okay, so see, the concept is very clear. Nuclease, that means these are the enzymes present inside the ribosome. Ribonuclease are the nucleus of the of the lysosome that can act on the RNA molecule. Okay, it acts on the RNA molecule, therefore it is called as ribonuclease. Okay, then the enzyme which can act on the DNA, which can cut the DNA, which can break the DNA, those are called as deoxyribonuclease. Am I clear?
Yes, ma'am.
Okay. Then the phosphatase, that means they can act on the phosphate esters. Then lipases, okay, so this can break down the lipid molecule. Then proteases, it can break down the protein. Okay, it can break down the protein. Then glycosidase, which can act on the which can break the polysaccharides into monosaccharides. Okay, and sulfatase, which act on the sulfate bond. Okay, so these are the enzymes present inside the lysosomes. So these are the function of lysosomes. Okay, this is the function of lysosomes. The first function is digestion of external particles. Digestion of external particles. Okay, then digestion of intracellular substance, which is called as autophagy. Cellular digestion. Okay, they help in the process of digestion that take place inside the cell. Extracellular digestion too. Extracellular digestion means by when these digestive enzymes are released, okay, they cross the membrane and they digest the things outside the cell too. So that is the extracellular digestion. Sperm penetration, then osteogenesis. Acha, listen. So we have to go a little bit details about this thing. Okay, since I have a class again from 9:45, so I need 5 minutes of break. Okay, so I shall discuss the topic in my subsequent class. In the meantime, I will give you the study material. So you go through the study material and we just discuss. Tumalke pohiba and we will discuss. If there are some doubts, that will be clear. Okay, these are not so much complicated thing, but again let me clear this thing that I cannot finish in by 5 minutes. Okay, so we will discuss in our subsequent class about this function along with you, not only me, along with you. We will discuss this thing in a very proper manner. Okay, so by this I want to conclude here. If anyone has to say anything regarding this thing, you can comment now.